So this course, again, is offered by Professor Onur Mutlu, and I'm Mohammed Alser. So the role of this course is to cover the basics of genome analysis. We're not going to teach you all aspects of genome analysis, but we will teach you the things that you may need in your projects. And we will focus, since this project is about mobile genomics, where we will do the analysis in portable devices or in a portable fashion. So we will teach you the things that you need to understand, the speed, accuracy trade-off in using computationally lightweight heuristics. And why we needed computationally lightweight, you may guess it that because these portable devices normally have a lot of constraints about energy, speed, storage, and so on. So we might not be able to use the traditional or conventional algorithms. We might use some heuristics. That's why they might affect the accuracy of these analyses, or not necessarily the end results, but might affect the current results that are related to that step. So we really would like to understand these trade-offs. And after that, each student will experimentally evaluate these heuristics algorithm, observe their effect on the end results. So we already know that these heuristics might affect the results related to that step, but not necessarily affect the end results, which is the output of the genome analysis, or in another meaning, whether you have a variation that causes a disease at some location. And these are one example of the end results. So we will study these in the algorithmic level, evaluate them, not necessarily building a new algorithm, but at least you will have a couple of existing algorithms, then you will evaluate them and see if there's a trade-off that you can leverage to accelerate the things or to implement it in mobile devices, such as your phone, maybe portable tablets, and so on. We will discuss these in details, hopefully in the next week, where we'll discuss the projects that we are offering part of this course. And this experimental evaluation that you will carry on will give you the chance to have a hands-on project. So you will implement the things by yourself. You will implement one or more of these heuristic algorithms after evaluating their behavior, their accuracy, their speed in your smartphones. And hopefully by doing that, you will help the society by enabling on-site analysis of genomic data. So you are aware about the pandemic that we are facing these days, and you can see the need, for example, to detect the COVID-19 or the coronavirus rapidly. Imagine you have a flight, so all the passengers need to do this test before they board and when they land in the other country maybe. And they do this on a daily basis. So every flight or every travel might require that testing. So it will be very helpful if we can have one of these portable devices. So we already have portable devices actually to what we call it sequence your genome. So I'm not sure if you already studied the learning material that we assigned through the email I sent, but if you go through it, you might understand some of these terms. So I'm going to explain in the next few slides about sequencing, but we already have this machine in a portable fashion, but we don't have the analysis in a portable fashion. And this is what we are missing. And your goal is to enable portable genome analysis, which is after having this, the machine that is located normally in hospital or wet labs where they read your genomes, they will give you some data in text format. And this text format is a messy data. You cannot get any information about it. That's why we call it data, not information. And our goal is to convert this data, this piece of data, into useful information that we can leverage using, of course, a computational approach. So we are not related in this course at all in anything, in any fashion to wet lab. So everything we will be doing, just developing algorithms, implement them. That's it. All right, so what are the objectives of this course? So we have multiple components that are aimed at improving student basic knowledge in genome analysis. Again, this is dry lab. There is no wet lab. There is no taking blood sample and such and process it in the wet lab and do all this step. This is something as a pre-processing, you can consider it. What you will do is developing algorithms, implement maybe hardware or software, and implement it in your smartphone. That's all. Of course, you will learn technical skills in genome analysis and computer architecture. We will go as we need. So we don't have a fixed curriculum to teach you, but if you get assigned one of these projects that we will list in the next week, then we will try to teach you the best that you need for that project. So we have a very good expertise in this field from the supervisors that I'm going to show you in the next slides. And all of these supervisors will help you a lot in all of these projects. Of course, you will develop critical thinking and analysis when you face a problem, how you can solve it, how you can familiar yourself with such problems in implementing algorithm genome analysis. And with that, you are ready to carry out maybe research if you're interested in doing masters later on, PhD. So you are already familiar with some of key research directions that even ourselves are still working on them. And hopefully at the end of the semester, you will have a technical presentation of your project. We will help you on that part as well, how to prepare yourself, how to prepare the slides, and how you can explain the things as if you are in a conference setup. Okay, so what's the key goal of this course? We will teach you how to efficiently implement one of the key steps in genome analysis on portable devices. As you all aware about the prerequisites of this course is that we don't require any prior knowledge in bioinformatics or genome analysis. We will teach you all the things that you need for this, successfully completing your project. And we prefer to have a good knowledge in C programming language and programming in general. So if Python might be okay, we will see that. But anyway, if you really master one of the programming languages, then it should be great to switch directly to another programming language. So normally the syntax are same. You just need to learn a few differences, how to do the if for loop, the if conditions, the maybe switch cases, and all of these things. There are very small differences between Python and high-level programming languages. For C, there are a bit more differences as it is a low-level, let's say, programming language where you really play with the memory utilization and such. But we will see. We might tailor the projects we have based on the skills you already acquired. That's why we will have one-to-one meetings in the following weeks to discuss all of this in details. Of course, you need to have interest in making things efficient and solving problems, because I think that is straightforward. Of course, this is not about doing research. We don't ask you to build a new algorithm, for example, but of course, the problems are there. In any project you carry out, you will have some problems. It might be related to the software or the algorithm itself, or it can be very simple, how to install this software, how to deal with this phone, how to do installing a C program on smartphones, and so on. So you should be interested in solving all these problems. All right, so this course is offered by Professor Onur Mutlu, who's a full prof at ETH Zürich. He's been there since September 2015. He was a professor at CMU before that for about, I think, 7 years. He already joined industry, different companies with very high profile like Google, VMware, Microsoft, Intel, AMD, and we're still getting funds from most of these companies. And in general, you can contact Professor Onur Mutlu through this email, the Gmail, he preferred. And Professor Onur Mutlu is leading the Safari research group, and oops. Okay. Can everyone still see the slides? Okay.
Yes,
good.
we can.
Great, thank you. So what we are doing in Safari Research Group, we have different research interests, starting from computer architecture to computer systems, hardware security, bioinformatics, and developing systems for all levels of the compute stack, from memory, caches, storage, the CPU itself, hardware security, and so on. Okay, so we have a good team here for this course. We have me as a lead supervisor, and we have a supervisor. I will be a supervisor as well for a number of the projects. And we have Dr. Juan Luna, who's already with us, I think. Yes, he's with us. We have Jeremy. Yes, Jeremy also here. We have John Fertina. Jeremy and Jan are doing PhD in bioinformatics as well as computer architecture. We have Damla, Nika. We have a number of other students that will help you carrying out your projects. We will extend, I think, this list in the coming days based on the projects that we have. And all of these has very good knowledge in genome analysis. They already have good papers in this field. So I recommend all of you to go through Safari web page and go to the Safari group or the people and start reading the research interest of each of these supervisors. Maybe scan their paper, their recent paper, and just get familiar with what they are doing. Okay, so what we are expecting from all of you, of course, the attendance is very important, such as these meetings or one-to-one meetings that are related to the project. It's very good to cope with these meetings, such as you do very good progress throughout the semester. So by the end of the 3 month or by the end of the semester, you will have a complete project. We assigned 4 learning materials. I think 3 of them are videos and the 4th is a paper, although 2 of them are papers, but they already have videos. So you can watch these videos and that will be much easier for you to understand the things. I hope all of you start studying these learning materials because we need to have it ready before the next week. And the next week we will present the projects, and to understand them, or actually to know the challenges or to know the details about each project, you need to handle at least the introduction to genome analysis. And as I mentioned, each student will carry out a hands-on project that you will implement it yourself. You might build, implement, do some coding, design, with a close engagement, of course, from the supervisors. You need to be ready for participation. Please ask questions whenever you don't understand anything. You need to contribute with ideas, thoughts. You might need to find alternatives, how you solve these problems. I face this problem. Please don't stop. Don't keep silent for a week and then show up to the supervisor. Please use the email. We have the mailing list. We will talk about other communication platforms such as Piazza. So try to find a way to contact the supervisors and don't please keep silent. It's very good to maintain a very good progress over weeks. Of course, to understand some of the things that you need to solve, you need to read relevant papers, how they have solved some of these issues in similar or existing projects. So this is something you don't have to do it, but of course, if you do a very good progress in these projects, that will motivate us to help you to get your project published in good venues. It can be the best conference or journal in bioinformatics or computer architecture. And this is what Professor Onur Mutlu mentioned, that yes, we will try to do it for maybe all of the projects if we have a very good progress. We will try to help you a lot in writing and how to submit it and the results, and maybe we continue over your project for the following month after the semester until the paper is in a good shape to get published. And that will help you a lot if you're interested in academia or in other aspects of life. Do we have any questions so far from the students? Anything not clear about the logistics of the course, requirement, expectations? Okay, seems everything's clear, hopefully. All right, so as we mentioned in the email, we have already the website ready for you, and we will try to keep it updated. Of course, we will keep it updated with useful information for the course. And for now, we will use the email. So please check your email frequently for announcement. We will also have the Piazza soon. We will send you an email about this in detail. We will use it. It's, I think, much better than the email. We can keep updated about any questions we receive from your side or announcement, or maybe a problem that you'd like to share it with your colleague, with all students, rather than just asking the supervisor directly. So I hope everything clear until now for this week. And we will start actually to assign the projects in the next week. I prefer to meet exactly in the same time as today, and the next Tuesday, if it fits all your schedules. So please, you can open the mic, just mention if it is okay for you if we meet the next week in the same time as today.
It's okay for me.
Okay, thank you.
Yeah, for me as well.
Mm-hmm.
For me also.
Okay, great. So I'll fix the time. I'll send you an email about the next week. What we will do in next week, we will have a list of projects. We will prepare their descriptions, and we will have the supervisors explaining these projects. And they will tell you more details about it, what we expect, what you should have as end results, what should be the implementation. And we will give you a chance to select a project, so you can show your preferences to 1, 2, or 3 projects, and then we will meet. So after you show your preferences, we will have one-to-one meetings, and we will try to understand your interests, skills, background, what previous projects you have been doing in the previous courses, previous semesters, such that we can really match the projects to your skills and interests. So it's very important that all of you succeed in completing the projects. That's why this step is very important to us and to you as well. It's very important that you study the learning materials before our next meeting, so that you can really understand what are the challenges with every week, so with every project. So in these projects, they have totally different objectives. Don't expect that you will do the exact same thing as your colleague doing, but we will have totally different projects that each one of them targeting a very specific step in genome analysis pipeline. So in the genome analysis pipeline, we have many steps, and so you can expect that these projects are really different from each other. And that's why you need to study these learning materials to learn more details about every project. Some of these projects, by the way, if you attend the Professor Mutlu lecture, the first learning material that we assigned, some of these projects are very related to the things he presented in the presentation. And the second one, which is the survey paper, if you still remember, from the learning materials that we assigned, also talking about many steps in the pipeline. So these 2 learning materials is a must. You have to study them. So that's all for the logistics. I hope everything is clear. We will try to give you more details about our next meeting, how to select the papers, how to yeah, choose the project that suits you. And we will discuss about the one-to-one meetings. We will have different schedule for this meeting between you and between the supervisor or between one of us, basically. So just please wait for our emails to get more instructions. Do you have any questions? Okay, so we have about 30 minutes. And with that, I would like just to refresh your mind and give you just a brief introduction to genome analysis without details, actually. So what is genome analysis? We've been talking about it for the last maybe 10 minutes or 15 minutes, but we didn't introduce it actually. Okay, so in genome analysis, our goal is very simple. So we want to have the complete sequence of ACGTs in DNA. And what are these ACGTs? So you might already have taken a biology course where you already know that our DNA or the genomic sequence in our cells is contained of these 4 chemical molecules: adenine, cytosine, guanine, and thymine. And to get this full sequence, or this entire sequence of ACGT, if we already have it, then we can easily compare it because it's a text format. And as a computer science or a computer scientist or electrical engineer or any field related to computer where you deal with software and algorithms. So it's easy for you to compare a sequence of text or string rather than comparing something that you don't see with your eye, which is a blood, for example, or the cells. It's very small. You cannot see it and compare the content of it. So this is our goal. We want to get this genomic sequence, the full content, and start to compare it with other sequences or with other species, with other humans, and observe how this genome is different from the others, such that I have different phenotypes, different eye color, different hair color, or even new disease that they don't exist in the other species. Unfortunately, we don't have such a machine. The machine that provides you such a complete sequence of DNA. Okay, so what we have these days. So in 2003, we were able to announce the first copy of the human genome, which we call it the
Mohammed, you are muted.
Oh, sorry. Thank you, Damla. So in 2003, there is a project was called Human Genome Project, where we were able to successfully read the full content of the human genome. There are a few gaps in that content, but it was the very first effort in the world to have this draft of the human genome. And the human genome is approximated to be around 3.2 billion bases. And it took us 13 years to build that draft, and it cost about $3 billion. So you can think about every character that we read from the human genome cost us $1. So that was very expensive effort. But in the following years, we learned how to do the things in a better way, but we're still very far from having our goal achieved. So in these days, what we have is a machine that provides you short sequences of your DNA. So you can have these pieces, which are totally unrelated to each other. We don't have any information how we can connect them together to build the full sequence of our genome, which is 3 billion characters. But this machine provides very high throughput. They provide a lot of data, a large number of these short sequences in about 44 hours, maybe to 1 day, depends on the properties you choose for these data. There are a lot of different parameters that you can control to speed it up or slow it down to get more data. And that cost us just less than $1,000 just to do this wet lab process, which we call it sequencing. So basically, we get the blood sample, we send it to the machine that we call it a sequencer, and the sequencer will give us the read file, which we call it FASTQ format. And these reads contain these little pieces of our genomes. And our goal is to perform read mapping to connect these pieces together and map them to another reference, exactly same as we do in puzzle. So if you have a puzzle, you have large number of pieces, you need the full picture to see it, and then start matching these little pieces to that full picture, such that we said, okay, I think this little piece can be located in this place according to the reference. So same thing we do here. We have a reference genome that we created in 2003, or we updated later on. And we start comparing to that. And if there is a very similar region in that reference, then we said, okay, this little piece coming from the sequencer, it might be generated from that location because they are basically similar. We keep doing this for all these pieces coming from the sequencer until we stick all of these pieces to that reference genome, and then we can have the full sequence or the complete sequence of our genome. Of course, it's not as easy as it sounds, but it's very challenging because these machines, or what we call a sequencer, have a lot of different properties, totally different things. The read length, for example, is totally different. It can be short, it can be long, it can be 100 characters, it can be 1,000,000 characters. It can be accurate, it can be inaccurate. So some of these machines impose their own sequencing errors because, again, they are machines. They are not that perfect. So sometimes they don't read these bases correctly. So they give you something that do not exist in your genome. And for that, you need to do a lot of algorithms to correct that genome and do heuristics such that you said, okay, I don't think that you have that disease, but these are coming from sequencing errors, maybe. That's why it's very important to correct these errors, for example, before carrying or before coming to any conclusion, basically. And you can see the shape of or the size of these devices totally different. One of them hand-size shape, one of them can be sticked or can be connected to your iPhone, but we have also fridge-size sequencers, which is really huge. Again, all of these pieces coming from the sequencer, they don't carry any information about their location, their relationship with the other pieces, how we can order them, how we can sort them, how we can connect them to the reference genome, and so on. So you can think about a very, very huge puzzle that you'd like to solve. Like, let's say 1 million piece puzzle you'd like to solve. It's really very challenging. Again, I like this example from John Fortuna, where you can visualize the short reads and the long reads coming from these sequencers. So you can think if they are very short, I told you an example of machine that provide 100 characters pieces. So think about comparing these 100 base pieces to the reference genome, or comparing this large pieces. Same thing with the puzzle. You can have very small pieces and very large pieces. Which one will be easier to solve? Of course, the large one. But again, the large one has a problem of inaccuracy. So there is no which one better. There is no best. This one is short but accurate. So when I match it to the reference genome, I have certain level of confidence that what you have as differences is coming from the disease, not from the sequencing errors. But for the long, it might make my job a little easier by matching these large pieces to a reference genome. So exactly as if you are solving a puzzle with large pieces. But again, I don't have that much of confidence that these differences coming from the disease, but not sequencing errors. And again, there is no best here. Both of them are still used. And sometimes they use mix of these because each one has unique advantages. And this is your job, is to collect these pieces and try to build the vase again. Okay, yeah, I think we have 20 minutes. So these reads might come from single source, let's say the human genome. So we take this sample, which is in chemical format. We cannot understand it, cannot easily compare chemicals to chemicals and to see the differences. So we convert it into text format, which are the reads, and we cannot do better than that. And this is the current technology, just providing us subsequences of your complete sequence. And for that, we need to do what we mentioned is read mapping, try to pick piece and match it to this reference genome, and keep doing the same for all pieces until we get them assigned to certain locations, and then try to build a smooth, nice, complete sequence out of all these small pieces. But this is if we know the source, if we know that these pieces coming from this human or this cell. However, normally what we have, especially with COVID-19, they took just a swab from your body, and you don't expect with that swab to have a single species there. So you have human cells, you have bacteria, you have viruses, you have a lot of things in the microbiome world. It could be something that even we don't know. So in order to, we are able to generate these reads. Again, there is no problem with generating reads for different samples, but when you compare them, which reference you need to pick? I don't know what exists in these samples. So I don't know what reference I should pick. That's why sometimes we pick a full database, a complete database of references, and start comparing all of the generated reads to all of these references. And that is a very challenging problem because some of the reads will match to many of these references. And then I need to do some heuristics to calculate or to find a way to resolve these ties between the references. And this is what we have again in COVID-19. So it's very challenging. Although we might know the reference for the coronavirus itself, but the viruses, for example, they can change their DNA from time to time, or RNA. They don't have DNA. These viruses, they have RNA, which is a different version of the DNA, or unstable version of the DNA, basically. Okay, so there are a lot of challenges with read mapping. We need to find many mappings of each read. So as this read or these pieces, so in this cartoon picture, here is the reference genome, this one, and these pieces are the read. So you can see many of them. We have short, long, and each one matched to a certain location in the reference genome here, for example. But the same read might match here and there with the same number of differences, for example. So which one to pick? So basically, we need to report all of them sometimes, and then we do later steps to filter out some of these based on some metrics, some quality numbers, and so on. We need normally to tolerate small various differences or errors in these between the read and the reference, such that we don't match actually the read to the reference exactly. We need to allow some differences. For example, if it is Netherlands, it's okay. We can still match it to Switzerland, the two words, although they are different. They are different between each other, but we are looking for these differences, right? To know the cause of the disease. So it's very important to observe or to maintain these differences between the read and the reference genome. And we need to do this process in a very fast way because sometimes it might be life-threatening, such as I think about critically sick children that has genetic disease in the hospital and waiting to know the results that which disease he has or she has, such that we can provide good medication on time for that kid, or can be grown up, or can be for COVID-19 and so on. Yes. Okay. So one of the brute force algorithm, or one of the silly way to do read mapping, is just to pick the read and try to match it to all possible location in the reference genome. Just try to do sequential scanning. you keep scanning all location in the reference genome. You pick the first location. Does it match? No. The second location, does it match? Yes, maybe. But how many errors? Maybe 20 errors. Okay, this is too far from each other. Let's try the third location. Does it match? Yes. How many errors between them or differences? 1 error. Okay, this might be a good location. And we keep doing this until we finish the entire reference. Then we sort these locations based on the errors they have. And then we pick the one with the least errors because they might be closely similar to each other. We keep repeating this. But this is a very silly way of doing that. And it's actually computationally very expensive. Think about N, which is the reference genome length. It might be 3 billion for a human, can be 50 billion characters for plants, for example, banana tree and such. So it might be very challenging to do this step. And there are a lot of algorithms doing that. But even with that, the best performing algorithm, it's still 150x slower than the existing sequencing machine. So the sequencing machine are very fast. They provide you these small of subsequences in very fast speed, but still with our algorithm, we cannot cope with that heavy workload. So how to accelerate genome analysis? So there are a lot of profiling studies, and they found that 70 to 90% of the time spent in real mapping, you can expect it, is spent in just comparing the two strings together. See how many differences between the read and the potential location. You can expect these numbers because most of the things you are doing just comparing actually. And 98% of the location that you are checking on the reference genome, they are highly dissimilar. So if you keep, even with the most advanced algorithm, such that you don't really compare with every single location in the reference genome, but you compare with some of them based on some hash tables, some indices that you build first, even with that, you still have a high number of highly dissimilar sequences. And what we are doing to figure out the matches or the differences between two sequences is dynamic programming. So why we use dynamic programming? Because we want to find the best enumeration between two words, for example. And this is the only way or the only efficient way to find the differences between two words or two strings. For example, Netherlands and Switzerland, it might not be just only flipping a character or substituting that character, but also we can delete one of these characters or insert a new character. And this is actually what's happening in our reference genomes. So some of the diseases are caused by alteration in the reference genome. Some of these characters are deleted, some of these characters are newly inserted, some of them even substituted by single character, and that causes some differences. It could be phenotype, it could be something that you don't observe in the current generation. It might be through several generations, actually. And the problem with dynamic programming, I'm not sure if all of you study it, but in dynamic programming, what we do is that in each cell or in each entry of this table, we do some computation such that we will reuse it again in the following cells. So when we go to this cells, we go and visit the computation or the result we already compute in the previous cell, we reuse it and carry over through all the next cells. This way, it's very good such that we don't need to repeat the same computation again and again at each cell because we just get the result from the previous cell. But this creates a lot of data dependencies between the cells. So we need to respect the order. We cannot just compute this table in one shot, for example. Even if we have very fast machine, we still want to compute the first cell, then the second, then the third, then the fourth. Think about the sequence is 2 million long, then we need to compute 2 million by 2 million matrix, which is very expensive actually to compute. Okay, so the third problem with using this. So we need to compute the entire thing again, as I mentioned. It can be very expensive matrix to be computed. And to compute this entire thing, it's very expensive to wait until the end. Then you figure out that these two strings are totally different. They're highly dissimilar. So why I waste computing all of this until I find that the result, which is 5 here. And if you know dynamic programming, then in this kind of algorithm, you might find the result in the last cell, which is here represent 5. And 5 means that there are actually 5 differences between Netherlands and Switzerland as a word. Okay, so what are these differences? How many differences are we looking for? So some of the disease, again, they have single SNP differences. This is an example. You don't have to consider the scientific conclusion behind it or the observation. So you can observe here the blood pressure, for example, and increasing from 100 to 180. And these are different patients, different individuals. And you can think that when we read their genomes, we can observe some differences between them. And with this kind of studies, when we compare many genomes and then we observe the same disease or phenotype between these individuals, then we can easily figure out the genetic differences between their genomes. So see these differences, for example. Can we infer anything out of these differences? Can we say when it is always A, the blood pressure is low or high? Can we say anything about the SNP2, what we call it SNP? So SNP is single nucleotide polymorphism, or which is single variation, basically, as simple definition. So can anyone of you say something about these SNP2? Is there anything that you can correlate with blood pressure? Whenever we have G, is it low? Whenever we have A, is it high blood pressure? Dennis, Lars, Nicolas?
No.
Okay.
Much correlation.
All right, very good. So how about SNP1?
Lots of correlation.
Yeah, what you can say?
If you have C, there's a rather high blood pressure and low blood pressure.
Okay, great. So you can say, like, if you have C at this location and character of C at this location, then you probably have a high blood pressure disease, right? And if you have a T, you probably have a low blood pressure, right? Now, if we bring a new individual here, and without even measuring his blood pressure, just by checking his genome, we might tell him, okay, you have high blood pressure disease, or you have this disease, or you might already have the genetic for that disease, but it won't appear now. Maybe it will appear in your generations, and so on. So in this kind of disease is what we call single SNP disease, or disease that are caused by single variation. It might not necessarily, again, be a harmful disease. It might be just eye color or hair color. But there are some serious disease that are caused by alteration of large amount of your DNA. So think about autism or obesity. So in this disease, there is alteration of around 600,000 characters, which in exact 593,000 characters deleted or inserted. So if it is deleted, then you might have obesity disease. But if it is inserted or duplicated, then you might have totally the opposite disease, which is underweight. And these are the references for this paper showing these studies, how they discovered this large amount of variation between two genomes. So it's very challenging, this problem. You're not looking for single difference, for example. There are a lot of things going on. And think about COVID-19. This is the easiest example for what we call city-scale microbiome studies. In city-scale microbiome, you go just to the train station and swap the train or swap the ticket machine and so on. And for those machines, normally you don't expect to have single species or individual. You'll have a lot of human cells, a lot of germs, viruses, bacteria, and so on. there are a lot of studies in that, especially with the COVID-19 time. We can see a lot of studies showing that how you would like to track these pandemic and try to control them. So we can do that by swapping everything we have in a country or in a school or in a university. And this one example of these studies was conducted in New York, with 1,500 samples collected from all New York subway stations. The most important thing with this study is that they found traces of plague, and this plague is very life-threatening. Actually, it can kill people. So if they find it in a crowded place such as New York subway station, then this is very dangerous. There are a lot of things in the media about this story, too many papers coming out of that, and they call it as a failure of bioinformatics, because later on, when they used the traditional method to collect these samples from the environment, then they didn't find any traces of plague. So there was actually a false positive from the tool. So we developed an algorithm that tells the people, okay, you have a coronavirus here, but it does not exist in reality. And this is very dangerous. So that will tell you that we don't normally need just to develop fast algorithm. Our goal is not to always get the best performance, but the accuracy in this application is very important, because we are using it to develop medical needs, such as medication or protection, quarantine, and so on. So it's very important to develop fast and accurate genome analysis. And that's why we designed this course, such as we need to investigate first the trade-off between the speed and the accuracy of this algorithm before you are going to implement them in your smartphone. So it's very important to evaluate these things. And with that, we still have a lot of open questions: how and where to enable fast, accurate, cheap, privacy-preserving, exabyte scale analysis of genomic data. These are a lot of performance metrics that we should worry about. We already talked about speed and accuracy, but if we want to do it at scale, for example, for all population of Switzerland, then you want it to be cheap, actually. It doesn't make sense to cost people on daily basis $1,000 or before their flight to pay $1,000 to analyze their DNA. We want to protect their information, because if you read a genome of an individual, you already know what his kids or what her kids might have as a disease, or a potential disease, or maybe something that can block their health insurance or can cause a lot of troubles in their life. And there are a lot of research, even in our group, in Safari Research Group, we start doing genomic privacy. And we are focusing as well on developing a fast solution to do exabyte or genome analysis at scale. So this is related about architecture, but in this course, we will keep focusing on developing the things for smartphones. We are not going to develop new architecture, but this is what we have normally in computer or mobile devices, where we have the microprocessor or the CPU, then we have the main memory and the storage. And the sequencing machine is totally separated machine, where we need to move the data from the storage to main memory to the processor to process the things there. Then we store back the data there. And this is very inefficient to do it. There are a lot of energy issues with that, inefficiencies. And only the data movement, for example, you can see here, single memory request consumed up to 800 times more energy compared to performing a complex add operation. So you are wasting 800x more energy just to move the data to the processor to perform AND operation, for example. That's very inefficient. That's why in our group, we are developing also specialized solution. Sometimes we don't need to move the data from the main memory to the processor. We can attach some processing units that are specialized to do some specific step inside the memory, such that we store and we compute over these data. We don't need to move the data anywhere. And even you can do further by doing this processing inside the sequencing machine itself, such that you don't need to move data at all, or you reduce the data significantly. This is just an overview, very brief introduction about genome analysis and the things that we are doing. You will see a lot of details in the paper that we assigned to you and the videos. Please watch it. And if you have any question, please send directly to any of us. And I will send you more details about Piazza and the next step for the next week, as I mentioned. Do you have any questions? All right. Okay, I think we can finish the meeting at this point. See you all in next week. Thank you so much.